Welcome everybody to the November edition of the WK webinar. We are exceptionally lucky today because we've got Cory Simpson with us today from University of Washington. Before I give it an introduction into Cory, quick bit of housekeeping rules. All attendees' cameras and microphones are switched off during the webinar. Webinar will be recorded and it'll be uploaded onto the website so you can watch it later. Please don't record anything. There'll be an opportunity to ask questions towards the end of the webinar. Please use the Q&A function or the chat room and I'll keep an eye on it. And then please leave your feedback and any information, please contact us on research@debra.org.uk. Now, I'm not going to speak too much because you're all here to listen to Cory and not me. So Cory is a dermatologist and assistant professor in the Department of Dermatology at the University of Washington, Seattle. He joined in 2021 and he runs a specialty clinic for blistering diseases and built a lab focused on these disorders. His research program applies CRISPR gene editing and advanced microscopy methods to a human skin model with the goal of identifying therapeutic strategies for orphan blistering diseases. Cory is funded by the NIH and various charitable foundations and received a Young Physician Scientist Award from the American Society for Clinical Investigation and a Rising Star Award from the World Congress on Rare Skin Diseases. He currently serves on the board of directors of the Society of Investigative Dermatology, and his ultimate aim is to translate bench findings into bedside to develop therapies for patients. And on that note, I'm going to pass on to Cory, who's going to talk about engineering human tissue models of EB simplex to discover new therapeutic strategies. I'm going to stop sharing my screen and over to you, Cory.
Okay, so I think we should be sharing the right screen. Let me know if not.
I can see your screen, Cory.
Okay, good. So my name is Cory Simpson, as Sagar introduced me, and thank you very much for having me today. I'm really pleased to get to interact with the EB research community as well as patients or anyone else who's signed on today and is interested in hearing about our work. I am an assistant professor, but I'm also a physician scientist, meaning that I see patients, but also run a lab where I try to understand rare skin diseases and how we can make progress for treating them. So today I'm going to talk a little bit broadly about that, and I'll try to present a high-level view of what we're trying to do, but also give you some of the interesting findings that we've been able to to show lately in the lab. So, most of my clinical work focuses on blistering diseases. Mostly I see adults. We have a separate pediatrics department. So for the young babies and children that have EB, they're mostly handled by our children's hospital. But at some point, people with these disorders grow up and they end up transitioning to an adult dermatologist. And so I do see some of those patients in my clinic. But really the goal of my lab, as this sort of blue arrow shows here, is to take what we see in the clinic, the blisters on the patient, to try to understand them at the level of the tissues that fall apart in these disorders, the cells that are not sticking together very well in those tissues, and then the actual organelles or the tiny sticky surfaces that allow these cells to stick together in order to prevent the skin from blistering and rupturing. But we're really trying to use modern tools to take these understandings of very, very zoomed in pathology or disease and to use tricks like live cell imaging, like this shown here, where we can bring these structures inside of the cell to life and actually watch these diseases as they're happening. So these are some movies of some keratinocytes or skin cells that are stuck together by these little green dots between them that are essentially spot welds between the cells that hold them together. But we're really hoping to take these findings that we see in the lab to understand how does this disease happen and really where should we be aiming our therapeutics. So a couple of disclosures. We do have a patent we've applied for thinking about a new therapy for blistering diseases. I've done a little bit of consulting for some of the companies that are thinking about doing clinical trials into blistering diseases to help them design their trials so that they're more likely to be successful. And then I've been lucky to receive some funding from the Leo Foundation And I'll talk about some potential what we call off-label therapies, meaning they're not officially approved by the FDA or EMA, but unfortunately for a disease like EB simplex, there are 0 therapies that are FDA approved for that disease. So everything is essentially off-label. So this is my lab. I'm located in Seattle, so you can see the Space Needle there. And let's see if I can get my pointer pulled up here. Here we go. So Space Needle here, this is Lake Union. My lab is down here in this area. And I've been really fortunate to have a number of young investigators and more seasoned investigators who have taken a chance on a pretty new lab that's studying these diseases. So I have a lot of medical students and others who will rotate through the lab and really try to help us to make progress on some of these diseases that really could use some new therapies. So we've migrated over to blue sky in the wake of some challenges with the other platforms. So I like to start with this quote that really emphasizes the point that basic science research is still essential to advance medicine. So there's a Nobel laureate in 2019, Bill Kalin, who wrote an article on the Washington Post called "Why We Can't Cure Cancer with a Moonshot." Cancer is a huge problem. And what he was sort of saying is, the quote there says, "Now is not the time to abandon basic science in the false hope that we already know enough to eliminate the pain and suffering caused by complex diseases like epidermolysis bullosa." So we really don't know enough about the basic science of these diseases to really know where should we be aiming our therapeutic strategies. But he also said the most important scientific discoveries often began with an unexpected experimental result that a scientist then pursued. And I often say this to members of my lab: when you find something in a lab that's really not how you thought an experiment should go, sometimes that's really a clue that maybe the disease is trying to tell you something, and you should listen to that surprising result and really follow it up, and maybe we'll find something interesting. So the outline of my talk is to really give you an overview of epidermolysis bullosa simplex, how it presents in the clinic, the genetic cause, some of the mouse models that have been made to study the disease, but also to talk about our new 3D approach to making human tissue models, talk about some of the research challenges, particularly in EB simplex, and then also talk about some of the promising technologies that are out there and potential roadblocks that are holding us back. So we'll start with the first area here, thinking about what does the epidermis look like. So when we talk about blisters within the skin, this is the tissue here. So the epidermis is made out of a bunch of different cells that have to stick together to one another to form a tissue that can resist tearing when the skin is being pulled or pinched or stretched. And so this is made up of keratinocytes that really have to stick together quite tightly. And they do this by coupling their bones, the bones of the cell, the cytoskeleton, which is made out of keratins. Basically, the cell can link those together through these sites of adhesion that are called desmosomes. And those essentially function to weld the cells together so that they don't fall apart. And that's what goes wrong in a lot of these diseases, such as EB, in which we lose this attachment between the cells and lose the structure of the bones that make up these cells and give them strength. And that causes the epidermis to fall apart, or the outer layer of the skin falls apart, and it develops blisters, which is what we see on the patient. And this is what it looks like if you take a biopsy, where you have these spaces within the tissue that really shouldn't be there. These white spaces really should not be there. And that's what a blister is. The cells have severed apart, and that creates a blister. So I went to the University of Pennsylvania to learn how to become a dermatologist, and learned from these two outstanding scientists who ran labs but also saw patients with blistering diseases. And they taught me how to take care of patients with blistering diseases. But what I learned from them is that there are really two different types of blistering diseases. One is autoimmune, meaning that your immune system gets disrupted, and that's what causes the blisters. But there's also a family of genetic blistering disorders, of which EB is one of them, in which the patients are born with a problem in their DNA that leads them to have skin blistering lifelong. And what I learned is that we have really good treatments for patients with blistering diseases that come from the immune system. We can use a therapy like rituximab, shown here, to really put most of these patients into remission. But we really didn't have anything specific for most of the patients that have blistering diseases that were caused by an inherited reason or a genetic reason. So that really motivated me to study diseases that are caused by genetic problems, because we really have no good therapies for most of these disorders, although that's starting to change with some of the recent approvals for dystrophic EB, and I'll talk about that at the end of the talk. So I want to focus today on EB simplex, because I think this is a type of EB that gets underemphasized. A lot of the progress that's been made and a lot of the research that's been done in the last couple of decades has really been focused on dystrophic forms of EB, or the deeper scarring forms, some of the cancers that arise from those types of EB. But my lab is really trying to make progress for EB simplex because we feel like this disorder has been understudied, and patients with this disease still do suffer greatly, especially in the early periods of life. So EB simplex is called when is caused by those skin cells, keratinocytes, that basically shear under mechanical stress. They rip apart. They can't hold themselves together very well. This often starts at birth. So the birthing process itself can be quite traumatic. Coming through the birth canal, you can imagine the skin is stretched and pulled, and that can lead to skin fragility and painful wounds even from birth. But it can also affect the inside skin, sort of the skin of the mouth and the throat. So it can cause issues with feeding and lead to malnutrition in young neonates. This disease was linked to mutations in the bones of the keratinocytes, the keratins, which make up the skeleton of those skin cells. And so there were some brilliant scientists, Pierre Coulombe's group and then Irv Epstein's group, that both published that these mutations within keratin genes seem to be the cause of EB simplex. So these studies were published in 1991, and they found that there were two different types of mutations. So you could have a dominant mutation, which means you only need to inherit one mutation in the gene to get the disease. Or there were also some really rare cases where both genes were damaged, and that led to total loss of these structural bone proteins that allow the keratinocytes to have strength. In adulthood, so this is largely, it's the worst in childhood and in the neonatal period. Adults can still have active disease, but the phenotype or the disease presentation tends to improve with age, and we really don't understand why that is. Why is it more severe in children and less severe in most adults? And that leads us, and me in the lab, to think, well, there must be ways that these cells have to compensate over time as a patient gets older to prevent the sort of more severe blistering that tends to happen in the early years of life. But patients as adults that I see a couple in my clinic that still have blistering that's related to, footwear that doesn't quite fit right, or manual labor, things like this that can actually induce blistering, and that can still be quite painful and quite limiting for patients. But as I mentioned, these studies came out in 1991, so we've known the genetic cause of EB simplex for more than 30 years. Yet there are still no FDA-approved therapies for EB simplex. And my lab has really taken this on as saying, well, that's not really okay. We need to do better than that. If we know exactly what causes these diseases, surely we can do better at finding new therapies for patients who have these disorders. So one of the first things that was done shortly after the disease was found to be linked to these keratin problems was to make an animal model using mice. So this is what scientists tend to do. They tend to try to replicate the genetic problem in a mouse as a model to show us what's going on in the human disease. So this was done in mice. But the problem was when investigators targeted the keratin 5 gene, which is one of the two that's mutated in the most common form of EB simplex, the mice died very early in life. They had blisters on their skin when they were born. They often died very early because of dehydration, trouble feeding. They just didn't survive. And so that really is a problem when you're trying to use this model to study the disease. If the mice really don't survive for very long, you don't have much of a chance to understand how did the disease happen and how might we reverse it. But you can see in these studies, this is from 2001, so this has been almost 25 years ago. And again, this is sort of stalled research in this field where we really haven't seen a lot of progress since this mouse model came out. What you can see in these sort of fancy scientific diagrams is that keratin 5 protein here is gone. So they've removed it. So that says that it worked. But what's interesting is that this other protein called keratin 6, which is kind of the nearest relative or cousin of keratin 5, actually got upregulated in the mice that didn't have the keratin 5 protein, which again leads me to propose that maybe there are actually native mechanisms inside of the cells that maybe they could turn on a different keratin to replace the one that is not functioning properly. And that's where some of my research is trying to find new ways to do things like that, to use native mechanisms that exist inside of our cells to tap into those to really fix the problem in something like EB simplex. But this also led us to ask, well, if the mouse really doesn't work very well for this disease because it causes the mice to be so fragile that they died shortly after birth, could we replicate this disease using a human tissue model instead of relying on an animal model like a mouse? So now I'll talk about some of the new 3D human tissue models that we've made, which I hope will be useful for the EB simplex community to make some advances. So we've used a technique called CRISPR-Cas9, and you may have read a bit about this recently, but it's basically a DNA eraser tool where you can essentially go in and change the DNA and replace it with whatever you want to edit in. So it's essentially like a typewriter that allows you to type what you want to go into the DNA. So this is being used as a tool to modify genes within cells to basically replicate what a patient might have or to replicate a disease state. And so we've used this in a couple of different blistering diseases that are not EB simplex, but now we're trying to take those technologies and move them over to study EB simplex. So this was our paper from a couple years ago. I'm using this technology to make mutations in a gene that causes a blistering disease called Dermatology disease, also a very rare blistering disease that tends to present in adults. So when we use this CRISPR system, we can target the ATP2A2 gene, which is what is problematic in the Dermatology disease, in keratinocytes, which are the skin cells, and we can basically take away the proteins that are missing in these two different diseases. So this is for Dermatology disease, and this is for Hailey-Hailey disease. These are two other blistering disorders that I see in my clinic that do not have good therapies either. So we were able to use this essentially a molecular typewriter to erase the genes for these two diseases inside of skin cells, which are the most relevant cell type to study for these diseases. But then what's interesting is that we can take these diseased keratinocytes, the keratinocytes that lack the same gene as in the patients, and put them into a 3D model that regrows the epidermis or the outer skin tissue within about a week or two. So this is really exciting because it gives us a piece of real 3D tissue of skin in the lab that we can then screen for, well, can we find new therapies that could possibly fix some of the problem in these blistering diseases to sort of put the pieces back together again to stop the blistering. So this is really exciting. And we've also tried to use this technique in a cell type called iPSC, or induced pluripotent stem cells. So these are essentially a special type of human stem cell that we can reprogram with the same mutations that cause these blistering diseases to use them to essentially replicate the disease in a stem cell that then we can grow into essentially a whole piece of skin in the lab, which is really exciting. So we've done this to replicate a couple of other different diseases like I showed you, and I just want to show you that this technology works quite well. So if we target this gene called ATP2C1, which is missing in Hailey-Hailey disease, a blistering disease, this is what happens if we take a picture of what the skin looks like in a cross-section. So if we sort of cut across it, what you see is similar to what I pointed out before. There are these white spaces within this sort of purple and pink tissue that really shouldn't be there. So this is called splitting of the tissue or acantholysis, and that's exactly what happens if you take a biopsy from a patient who has this blistering disease. The skin cells split apart from one another. They can't endure the mechanical stress that happens to the skin, and they rip apart, which is essentially creating a blister. So what we've done is to be able to create a model of these blistering diseases in the lab that allows us to replicate the 3D pathology or the tissue disease of these patients in the lab, which is really exciting because then we can test out therapies in the lab before moving into a patient to make sure that things are safe and that they're effective before we really try it out in the clinic, which is a really important advance. So one of the ways that we do this is to look at all the genes inside of these cells to understand, well, what's changed inside of these cells? Where should we be directing our therapies? Otherwise, we're kind of just shooting blind and we really don't know. Where should we be trying to find new therapies for a disorder like this? So we've been able to take some gene sequences out of a disease like this and to find some targets here in red that we might need to aim our therapies at for these diseases to really make progress for patients. So could we apply a similar approach to model EB simplex as we've done with these other blistering diseases, which is what my lab is working on now? So this is Karina Schmidt, who's a graduate student. She's working on her PhD in my lab, and she's really the one who's driving this project. We collaborated with a lab at the University of Michigan, the Good Johnson lab, to target the disease keratin 5, as shown here in red. And so if we use this same sort of DNA eraser tool to mutate the gene keratin 5 in these same skin cells, can we do that? And would that be a model for EB simplex? So we were able to sequence the DNA of these cells and confirm that we were able to get rid of the keratin 5 protein, which is the protein that becomes dysfunctional in patients with EB simplex. So where the yellow arrow there, it just shows that we're able to remove this protein from the cells, from the skin cells, which really starts as our base model for trying to replicate this disease in the lab so that we can figure out better ways to treat it. So if we take those cells that are now missing that sort of keratin protein that allows them to make strong bones, and we actually look at what their bones look like in the cell, this is what it looks like. So if we stain the cells for keratin, all of these little circular structures are nuclei of the cells. So this is a sheet of cells grown together. What you can see is in the controls on the left, the cell bones basically stretch out to the periphery or to the edge of the cell, and this is what it's normally supposed to look like. It's supposed to be the whole cell that's kind of filled up with these strong bones that make the cell strong. But in these 2 KO or knockout lines, we call them because we've knocked out the gene, what you see is that the cells look quite different. The bones of the cells seem to be weaker. They seem to collapse in around the nucleus of the cell, which are sort of these stronger ring-like structures that the yellow arrows are pointing to. And this is exactly what's seen if you take skin cells from patients with EB simplex and you grow them in the lab. Their bones don't properly stretch out. They tend to collapse and fracture. And this tends to be what we see in patient cells. So we think we've got a pretty good model for replicating the problem that we see in actual patients who have EB simplex. So these bones of the cell, the intermediate filaments, are connected to these sites of cell-cell adhesion, which is what allows the cells to stick together to one another so that they don't tear apart when the skin is pushed or pulled or pinched or scratched. So we started to ask, do these cells actually have impaired integrity? Can we show that they mechanically are weaker in their ability to stick to one another? And so we use a specialized technique in which we grow these cells in a petri dish, and then we lift them off once they're forming an intact sheet of cells, a sheet of like 2 million cells that we lift off of the plate. And so we have all of these little cells that have their nuclei here, and they're all attached together. And this is what it looks like in the lab if we take a picture of it. It's a little sheet of skin, essentially, that we grow in the lab. And then we can apply mechanical stress to these tissues to say, do they break apart more easily, which is what happens in a disease like EB simplex. So if we apply mechanical stress to these sheets of cells, do they break apart? And they do. So we can actually count the pieces of cell sheets that remain after they endure this mechanical stress as a proxy for how strong are these sheets of cells. So this allows us to apply a quantitative measure in a fairly cheap and repeatable experimental approach to show, do cells that lack the same gene in EB simplex, do they fall apart more easily? And could we find a drug that essentially reverses this and allows the cells to stick together more strongly and not fracture in a very simple model like this? Could we actually use it to screen for novel drugs that could actually make the cells stronger to overcome the problem that is what is driving the pathology in a disease like EB simplex? So that's one thing we're pretty excited about. But we've also taken this out to the tissue level where we grow a little raft made out of collagen, and then we put keratinocyte, the skin cells, on top of it. Then we float that raft on top of the liquid medium, which exposes the skin cells to air. This is enough to drive them to form multiple layers, which is what the epidermis is, the outer layer of your skin that you can touch with your finger. That's a multiple layered structure of keratinocytes, or these skin cells that essentially form multiple sheets of cells. And if we take, again, a cross-section where we're sort of cutting through and looking at them from the side, this is what the typical cells look like, the control cells. You see the keratinocytes, they form this multi-layered epithelium. But then if we have our cells that lack keratin 5, this is what they look like. They're very fragile, they're very small, and what you can see is that the lower layer cells at the bottom of the culture really sort of get torn apart. And this is exactly what the pathology looks like if you take a biopsy from a patient who has EB simplex. You can see where the arrow's pointing here, these poor cells in the basal layer, the lowest layer here, seem to get torn apart. So we do think that we're actually replicating in 3D the tissue problems that actually occur in a patient who has EB simplex. So we think that this could be used as a model to find new treatments that might restore the integrity of this skin tissue to allow it to form more strong connections and to form stronger cells that don't tear apart so easily with mechanical stress. So this is really exciting to my lab. And we're trying to use this model to really advance the way that we study EB simplex in particular. So one of the interesting things that we found, and this goes back to that original quote that I presented about, sometimes you find something unexpected and you probably ought to follow it up because maybe science is trying to tell you something exciting. So what we found is that while most of our diseased cell lines, these KO cell lines, form these sort of thin tissues that were very fragile, we found that some of them were able to compensate somehow spontaneously without us doing anything to them, and they were able to form epidermis or skin tissue that looked much better than what their counterparts did, who also lacked the gene. And it wasn't because they turned the gene back on, it was gone still, but they had figured out some way around this problem of not having keratin 5. And we want to figure out, well, how did they learn how to do that? And can we actually tap into that mechanism to allow for a new treatment of patients? So one of the ways that we think that they did this was by turning on a different keratin. So the skin cells have a lot of different keratins that they express. There are 54 different keratin genes, and keratin 5 is just one of them. So they're missing one of them, but they have access to a whole bunch of different keratins. One of those keratins is called keratin 10, KRT10. And what we found is that the expression or the level of this protein actually went way up in these compensated knockout cultures. So it suggested that maybe these cells that lack keratin 5 might have a way to sort of sneakily turn on a different keratin to serve as a replacement if they don't have enough keratin 5 to resist mechanical stress. And so that's pretty exciting. We were able to show this on a Western blot shown here, where the darkness of these sort of black blobs tells you how much protein is around. In some of these knockout cells that were able to compensate, we saw that the level of keratin 1 and keratin 10, which are partners together, they were increased in these cells, suggesting that they may have an ability to actually upregulate other keratins to replace the one that's not functioning correctly, which could be a really important way to possibly treat patients that have this disease. So normally we think of keratin 5 and keratin 14, which are the sort of keratin genes that are problematic in EB simplex. They're normally in that lowest layer of cells down here, which is why that's where the cells sort of split apart. But interestingly, if the cells can escape that lowest layer and move up into the second layer, that's where they start expressing a protein called keratin 10, which is shown here. So in the upper layers, they start expressing a different keratin called keratin 1 and keratin 10. And these layers don't seem to fall apart as easily if you take a biopsy from a patient who has EB simplex. So this led us to wonder, well, can we actually leverage these endogenous compensatory cell mechanisms that are downstream of the loss of function of keratin 5 to halt skin blistering? So maybe cells are smart enough on their own to actually figure out how to get around this problem of not having a keratin 5 gene that's actually functioning. So maybe we can tap into that and figure out how are the cells doing that, and can we actually turn that on intentionally, not just by chance, but use a drug to actually induce that phenomenon to allow the skin to strengthen itself inside of a patient who has this disorder. So that's something that we're really interested in looking for, are drugs that can turn on keratin 1 and 10 as a potential replacement if you're missing keratin 5 or 14 or it's not functioning correctly. Okay, so I want to talk now about some of the research challenges. We're very encouraged by those results, but of course there are always challenges about, well, how do you get from that model to finding something that might actually help a patient? So how do we find the key targets that are hidden in mountains of data? So right now with scientific technology, we can generate huge amounts of data from even rare diseases. So even from just a few biopsies from patients, there are all these different what are called omics techniques, which basically means you're looking at all of the genes in the cells, all of the proteins in the cells, all of the metabolites within a cell, and we can really characterize these things incredibly well with mountains of data. But the problem is, how do you find the needle in that haystack? The saying is, how do you find the needle in the haystack? Meaning you've got all of these possibilities, all of this data, but how do you search through and find the one thing that you're looking for that might be something that we call a druggable target, or something that we can actually aim a therapeutic at for benefit in patients? So one other question is, well, is it really going to be as simple as that? Are we really going to find the one thing in all this mountain of data that's going to be relevant for treating EB simplex? Is it going to be one drug that might actually help patients to have stronger skin and to resist blistering? Or are we possibly going to need combination therapies? And that actually gets even more complex. But there's been a lot of progress made in these inflammatory diseases like psoriasis and atopic dermatitis, where by having these huge maps of data, investigators have been able to find these key signatures that tell them this drug might work for this disease, whereas this drug might work for this disease. So we're hoping that we can use that for a blueprint for a disease as rare as epidermolysis bullosa simplex to really find where should we be aiming our therapies? Where's the needle in this haystack of data? So we've done kind of our own miniature haystack of data, which is RNA sequencing as shown here, where you get these little blue dots are all the genes that are down regulated in the cells, and all the red dots are genes that are up regulated in the cells. And this has pointed us to a number of exciting possibilities that might connect a structural protein like a keratin to something that's a signaling pathway that might actually be targetable with a number of different drugs. So some of the recent scientific evidence suggests that these physical structures inside of cells, like the adhesive structures or the bones of the cytoskeleton, that they might actually be connected to signaling pathways like some of the ones I've shown here. And the cool thing is we already have drugs existing that might target some of these signaling pathways, suggesting that we might be able to borrow those drugs from other diseases to use them for a disease like epidermolysis bullosa simplex, which doesn't have the benefit of having large populations of data from a vast array of patients because it's a pretty rare disease. So for example, I'll show you here that we were able to use a drug called trametinib, which is shown here in the magenta text here. This is a MEK inhibitor that's used in cancer therapies. And so when we found a signature in our data that suggested that this pathway might be overactive in patients that have EB simplex, this led us to test it in our model. So here's just showing you these protein blots that show you that the keratin 5 protein is gone in our knockouts. And treating with trametinib, this FDA-approved inhibitor, doesn't bring back the keratin 5. That's not possible in these cells. But what it did do was to increase the expression of proteins called keratin 1 and 10, which I mentioned before. And it also shut down the signaling pathway, which we can measure shown here. So in the magenta boxes, what we've shown is that if you compare lane 2 to lane 3 and lane 4 to lane 5, the blobs of black here are bigger for keratin 1 and 10, which is important. So this tells us that in these cells that lack keratin 5 that are kind of replicating EB simplex, we're actually able, with a simple drug that's already approved for a different disease, we can turn on this alternative keratin scheme that might actually allow the cells to replace their dysfunctional cytoskeleton with an alternative cytoskeleton. So it sort of gives them the ability to swap in a different keratin since the keratin 5 cytoskeleton is not working well for them. So that's pretty exciting. And it actually seemed to increase the strength of cell sheets. So if we look at these cells and look at their junction or their ability to stick together, you can see the green staining on the bottom, which are the drug-treated cells, seems to be stronger. So they seem to be forming stronger attachments to one another, which probably makes them stick together better. But we can test that with our mechanical assay, where we take these cells that lack keratin 5, we subject them to mechanical stress on the top in the controls. They actually break apart, as we saw before. But if we apply this drug called trametinib, we actually saw that they stuck together quite a bit better. So that's encouraging. If we can use a simple drug that's already been approved for other diseases to allow these EBS-like cells to stick together more strongly, could that actually be onto a possible therapeutic strategy for these disorders? Okay, so, is this actually a viable therapeutic candidate? So my lab has studied a disease called dairy disease that I mentioned earlier. And what we saw that is if we took biopsies from patients with that disease, we saw a similar upregulation in this pathway, as is shown here by the increase in the green staining. And someone took this idea and actually used it in a patient who had a very severe variant of Darier disease and used trametinib, which is what we use in our research paper, to treat this patient who was really desperate and severely affected by this disease. And of course, this is one patient. You have to do bigger studies to actually replicate this to find, first of all, is it safe and is it effective in all patients or is it just certain patients? But for this patient, someone took the research from our lab and actually used it to treat a patient. That was incredibly exciting for us. And it also told us that this drug trametinib might be viable as a therapeutic drug for something like EB simplex. Could we borrow this drug and use it for a different rare disease, and would it work the same? So could MEK inhibitors be so-called repurposed for keratin 5 loss of function to halt blistering that we see in EB simplex? And so that's something that we're excited to explore, and we're currently seeking grant funding to do that type of research. So finally, I want to talk about a couple of new promising technologies and some potential roadblocks that exist, because there's always, two sides to the story, of course. But I've tried to be optimistic about some of the stuff that we found, but we like to think down the road, how do we actually get these things into the clinic? And are there going to be more advanced tools that we should be using to properly research this disease? So one of those is to build upon our base model, which sort of took away the whole keratin 5 protein. Could we use this system to actually build patient-specific models? So I showed you that by taking out the whole protein keratin 5, we were able to replicate the pathology of EB simplex. So we could show you that the cells fall apart. If they form a tissue, the basal layer of the cells basically shear apart under mechanical stress. But instead of removing the whole keratin gene, could we perhaps design this sort of CRISPR eraser to insert a specific mutation, which is what a patient might have. Something like this R125C, which is a pretty severe variant that's found in the keratin 5 gene. So could we actually replicate that gene that a patient with EBS has in this keratinocyte cell type to then produce tissues, to essentially produce the same tissue like what is in that patient in the lab, and then use this as a testing ground to say, well, for that patient's specific pathogenic variant that's in their DNA, can we replicate that in the lab, grow some pieces of tissue that are essentially modeling what that patient has, and use those pieces of tissue in the lab to screen new drugs that could actually lead us to find what would be a personalized therapy for that patient, which I find incredibly exciting. Science like this a couple of decades ago would have been just science fiction, but now we actually have the tools that allow us to build these unique models, to edit the DNA exactly how we want to, and then to potentially screen for personalized therapies for patients, which I think is really incredibly exciting. But how do we actually move from the bench in the lab to the bedside of a patient? How do we get drug companies interested in EB simplex as a very rare disease that's often not the most easy disease to generate a new drug for, that has to generate some sort of profit for a commercial entity? So could we possibly fund repurposing trials for drugs that already exist to bring them over to EB simplex, which is one of the things that my lab is working on? So one of the questions is, do we already have the tools that we need to treat EB simplex? Is there a drug that exists out there that we could borrow and use that for EB simplex? Well, there are thousands of drugs out there, right? So it's really impossible to try them all out one by one and sort of hope that they work. And that could be quite time consuming and quite dangerous and very inefficient. But one of the things that people have tried to do is to repurpose drugs that we're familiar with in dermatology, like biologics. So these are injectable drugs that target very specific parts of the immune system. So something like dupilumab or secukinumab might be effective for some aspects of the disease in EB simplex. But some of the research in other rare disorders like epidermal differentiation disorders or ichthyosis, which is a different skin fragility disorder, what the research has shown us is that just sort of taking any old drug and trying to use it in a rare disease doesn't always work. It might help some patients, it might not help others. So sometimes we talk about this, the analogy where it's like you have a hammer, so then every problem looks like a nail. But maybe the problem of EB simplex isn't a nail, and maybe we don't have the right hammer in our hand to really treat that disease and to get it back into a better situation where the patients aren't having wounds. But oftentimes what we have at our disposal is what we try first. So there are some trials out there to try some of these biologics, which is exciting, and it may really help our community, and that would be great. But is targeting these cytokines or receptors really the right strategy? If these aren't immunologic diseases caused by the immune system, do we have the wrong hammer in our hand for the problem that is EB simplex? Could we actually predict which patients might respond to some of these specific therapies, or do we need a totally different approach like some of the molecules that my lab is studying? For drug repurposing, so if there's a drug that already exists, that really is exciting for rare disease communities like the EB community, because it avoids some of the really expensive early phases of drug development. So some of the early parts of the testing you don't have to necessarily repeat because they've already been shown to be safe in patients and effective for at least one other disease. So that's really great. Might this make these therapies more accessible to more patients? That would be really great too, because they're already out there, they're already on the market, they've already been manufactured at scale. Can we sort of borrow them to use for a rare disease, which I think is very exciting. But then if profit is limited for repurposing drugs that now can't be patented for some specific indication, how do we actually motivate the people that have the money to do that research to actually do the clinical trials? So that's a potential roadblock, but something that DEBRA UK and other organizations are really trying to help academic investigators and others to partner to use some of these therapies in a repurposing trial. Finally, I want to mention that I think it's really cool that there are these designer new drug therapies that are out there. And I want to talk about some of them here in the last slide. So there's one thing which is therapeutic efficacy, which means, do these therapies actually work in patients? And you may have heard through some of the EB research community or patient communities about some of these really exciting new designer drugs that are essentially genetic therapies for patients with EB. Unfortunately, they're only approved for patients with junctional or dystrophic EB, which means we're still lacking in new therapies for the EB simplex community, which I think is a real problem. So there are some new things on the horizon. So viral gene delivery, which is what Vyjuvek is, that's approved for dystrophic EB. This would work in diseases in which you have a haploinsufficiency or you have a missing copy of a gene, meaning you just don't have enough of that gene. So that's not usually the problem in EB simplex. So we think that viral gene therapy probably won't work for something like EB simplex so well because it doesn't address the primary problem in EB simplex. There's some exploration now about nanoparticle siRNA delivery. What this allows you to do is to use a special form of RNA to sort of mute the mutated copy of the DNA or the RNA that gets transcribed. So this is really a useful technique if you have mutations like in EB simplex, where you have one mutation on one copy of the gene that causes what we call a dominant negative effect, meaning having that one copy with a typo in its DNA is enough to cause the other copy to not function well either. So this really could be something that might be effective for something like EB simplex, but the challenge is how do you make enough of this drug to actually treat the whole body surface? So in patients with EB simplex, especially early in life, they often have widespread blistering over their entire body. So how do you adapt a therapy like this that's pretty challenging to manufacture for larger areas of the body? But if you had more focal disease, like if it mainly affected your hands and your feet, maybe it would be more feasible for a disease state like that. There are now CRISPR gene editing vectors that sort of combine these two technologies together, where you take a virus that then delivers the CRISPR machinery to actually fix the typo in the DNA that could actually fix the problem, the root cause of a disease like EB simplex. The problem is you have to individualize that therapy for each gene variant that exists. So in EB simplex, there are scores of gene mutations that can cause the disease. So you'd have to design an individual therapy for each of those genetic problems to fix it, to basically correct the typo in a unique manner for those patients. And then finally, you may have heard about some of these gene-corrected epidermal grafts. So Zevaskin is this one here, where they basically take cells out of patients and reprogram and then fix the typo in the DNA in the lab, and then use that to grow grafts, skin grafts, that can actually be genetically corrected to then be sewn on to patients. And so this is actually being moved forward in patients now. It's been FDA approved, and it's really exciting because it takes the patient's own skin cells and then reprograms them in the lab and basically allows for them to grow personalized skin grafts that won't be rejected by the body. So this is really exciting, but it's also very challenging. If the whole goal is to basically re-sew on skin grafts onto the entire patient, you can imagine that that's very surgically challenging, would be very challenging on the patient, and has to be in a really sort of high-risk situation, which is why it was approved for the deeper sort of scarring form of recessive dystrophic EB. So finally, I want to mention therapeutic equity. So therapeutic efficacy is, do the therapies actually work? Therapeutic equity refers to who gets these therapies, who has access to them, and what are the ethics around a question like that? Are these new designer treatments, which are really exciting technologically and scientifically, but are they sustainable? What is their cost, and are they actually affordable to the patients who actually suffer with these diseases? Which patients and which countries actually have access to these therapies? So they don't seem to be equitably distributed across the world. There are many countries that are much more resource limited that simply are not going to have access to these therapies, which is a real problem when we're talking about trying to really benefit the entire global community of patients who have these rare disorders. And finally, I think we have to ask ourselves, well, can we partner with companies that are truly committed to the long haul for rare diseases? So now that these companies have gotten approvals for some of these other rare disorders, are they going to keep up the fight and really go to bat for our rare disease communities, and to keep these therapies in the pipeline, keep developing them, and optimize them for even other rare disorders that really haven't gotten the benefits from these new therapies? So can we take these therapies and really move them into other diseases that have not yet benefited from these technological advances? So with that, I'll give a summary that, we've seen, recent treatments that have been FDA approved for junctional and dystrophic EB, which is exciting, and that's great for members of this community. But we have not yet seen similar targeted therapies advance for EB simplex, and that's really where my lab and others are coming in to try to make new models of EB simplex and to find better therapies for this disease community, which I think sometimes feels a bit left behind when these therapies are really marching forward for other forms of EB. So using this sort of molecular eraser CRISPR, we made renewable human cell and tissue models of keratin 5 linked EB simplex. And doing these sort of RNA sequencing of these grafts, grafts in the lab, we've been able to identify specific molecular drivers that might be druggable with existing drugs that already exist, which is pretty exciting. But how do we take this back to the bedside? Can we actually translate the findings that we make in the lab into new therapies for EBS by repurposing drugs that are already FDA approved for different diseases? And that's something that we're working on now. Future directions for my research really are intended toward that last slide that I showed you about our work, trying to build patient-specific models to enable personalized medicine. So trying to replicate the problem of an individual patient or group of patients that have a similar problem in their DNA. Can we actually replicate that problem in the lab to really find a personalized therapy that's going to work for those patients? And then can we build upon our platform that we've developed for EB Simplex to perhaps uncover novel therapeutic strategies for other rare skin disorders, so that other rare disease communities are similarly benefiting from the scientific advances that we make. And with that, I'd like to thank our collaborators as well as sources of funding. So I have been lucky to be funded by the NIH at various points in my career. My grants have actually expired at this point, so we're now applying for new grants. But as you may know, in the United States, we're always struggling with how to properly fund the NIH to really drive this engine of biomedical advances for patients. And so I really try to talk to my patients in any group that I get in front of about how important it is to make sure that we have scientific funding for large institutes like the NIH to make sure that this research, especially in the rare disease space, goes forward to help patients. So with that, I'll be happy to take questions. And I really thank you for tuning in today to hear about our work and our desire to connect with the EB community. So thanks very much.
Cory, I just want to say absolutely amazing talk today. I've learned so much. I thought I knew EB Simplex, so clearly I need to listen to you a lot more. So we are now open for questions for the audience, and we've already got two that come in.
Okay,
so the
great.
first one is, thank you for a great talk. Has a therapeutic equity been formally defined in any declaration or written work?
Yeah, that's a good question. I'm certainly not an expert in that area of like medical ethics, that type of thing. But I think it is something we should consider as these therapies are moving forward. Really, who does have access to them? I think that's something that as a community where the strength come from binding together and in numbers, I think we do want to think about, the patients who don't have access to these therapies that are coming out. So I think that's an important thing, but do we actually have a formal definition of that? Not to my knowledge, and I think that's something that we probably should address as one of the priorities of a research community is to make sure that the therapies we come up with have at least a pathway or a feasibility to be accessible to more patients than just sort of the privileged few that might have access to these. I mean, countries like the US or the UK, for example, which are generally just have much better access to advanced therapies like these.
Yeah.
Great point.
Okay, questions are coming through thick and thin here. So the next question is, they might have overlooked it, but was the observed compensatory effect in the 3D knockout model spontaneous?
Yeah, so we're working on that now. So I didn't give you the whole answer just yet. That's really what my lab is trying to figure out right now. It's like, how in the world were they able to, whereas their counterparts have all this fragility, how in the world were these cells grown in the lab able to figure out a way around that? So that's really what we're working on now. We're using primarily RNA sequencing to find out what's the code, like how did they get around this problem? The first sort of, logical approach was, well, maybe they upregulated a different keratin, and it does look like they express more keratin 1 and 10, which we're proposing might be a replacement for keratin 5 and 14. And that's really why we're looking at this MEK pathway to sort of turn on those two genes as a potential replacement. But we really have only scratched the surface of that data set to understand exactly how do these keratinocytes compensate and allow themselves to form a much more intact epithelium. So that's something that we're really interested in figuring out.
There's a follow-up question. Was there any morphological changes to these K10 upregulated cells compared to normal keratinocytes?
Yes, so they looked what we would call like hyperdifferentiated or hyperkeratotic, where they started to accumulate more sort of dead layers on top. So I thought that was interesting, suggesting that maybe they're triggered to differentiate or to mature more quickly. And so that may be one reason that they got to look that way. And this may be what we see on patients who have EB simplex is that often their palms and soles will thicken over time. And that's usually because they've accumulated thicker dead layers on top as sort of a secondary way to protect themselves from outside forces. So this might explain how it is that adult patients later in life are able to get, some sort of compensation to where their skin is not nearly as fragile as it was when they were younger. But that's still something to be figured out. But that was one of the morphological differences. The skin, the dead part of the skin on top, seemed to be a bit thicker in those patients, or in those samples.
Yeah. And then what's the regenerative potential of these K1 or K10 positive cells?
Yeah, so can they grow and differentiate just like the K5? We don't know yet. So one of the reasons that these cells get turned on is to sort of help with the differentiation process. So once they move into the second layer, they turn on this other family of keratin genes, and that may sort of take them down a non-proliferative route. But what we didn't really see in those cultures, it wasn't like the whole structure collapsed. So we think there's probably a therapeutic sweet spot where you can get enough keratin 1 and 10 to sort of mechanically bolster the cell without sort of triggering it into like a cell death pathway where it's going to march through and get to the end of its life too quickly. Because yeah, that could be a problem if you end up getting too thin of a tissue that now differentiates too quickly. That could be a problem too. So are we actually reducing their proliferative ability by turning on K1 and 10 early? That's something that we still have to look into. Great question, though.
Another question, completely different. So the caller has a niece who has a simplex and junctional mixed variant. They would like to know if there's any gene therapy that could be helpful for their niece at the moment.
Yeah, so it depends on what the specific mutation is in that patient. So if they have sort of a crossover, really genetic sequencing is the best way to figure that out, to know if you would be eligible for any of the FDA-approved treatments that are out there. So that's a question that has to be raised with your current treatment team. What is the specific problem in the DNA that is linked to the disease that she has? And would she qualify for one of these therapies that are out there? The one therapy called Filsuvez, which is the birch triterpenes, has been approved for junctional EB as well. So that could be used if she happens to have a genetic problem in the gene that causes junctional EB, she might be eligible for that. But the Zivaskin and the Vyjuvek have only been adapted for the dystrophic forms because they specifically are correcting the collagen 7 gene. So if you don't have a problem in the collagen 7 gene, those therapies really wouldn't work for that type of disease. Yeah.
I've got a question myself.
Sure.
And at the beginning of your talk, you mentioned that EB simplex is quite severe in children, and then over time it becomes milder and milder as you grow up. Do you think the role of K5 becomes less important as the older you get?
It's a great question. We don't have any evidence that somehow adults are able to correct the mutation that they were born with. They still have the same problem in their DNA, but something about getting older, you may have a different complement of keratins that's more or less important. Like I said, there are 54 different keratin genes. They all do probably slightly different things, but are they similar enough to kind of swap in? So that's one of the other arms of our research plan, is to try to see, well, are there other keratins that are really similar to keratin 5? Keratin 6, for example, is over 80% identical to keratin 5 at its protein sequence. So could that substitute in for keratin 5 for most functions? It may not be a perfect match, but could it actually be upregulated in these patients long term? I don't know that we actually have the data from that, but it might be something that we turn to the EB community and say, would adults be willing to get a biopsy to figure out what is it in their skin that allows it to stick together more strongly, generally, than a baby? So and every patient is different, right? But in general, adult patients tend to get better over time, which is great for them. But we don't know why that is. But it tells me that there must be some natural mechanism that the skin is able to adapt to that loss over time. So there has to be some way that it can get around that problem. And I think maybe our cellular model is revealing that might be true. And this might be a more feasible way to figure out what is that signature that allows them to kind of get around the problem that the patient was born with. But that gives me hope that there are ways to fix this problem. I think we just have to figure out what those natural mechanisms are.
Yeah. And then another question for me, temperature. You talk to a lot of EB simplex patients, and they will say their blisters are worse in the summer. Anytime the weather gets hot, it's a nightmare. And in the winter, actually, their blisters are a lot better. What do you think is happening there then?
Yeah, I think that's a really interesting question. This is where, like, when patients tell us things, we have to listen to them because they're probably telling us something about the underlying disease mechanism. They're probably giving us some clue. And so it could be that keratin proteins might aggregate more. So those sort of rings that we saw around the nucleus where the bone sort of collapsed, maybe that happens more at a higher temperature. That's a pretty simple thing that we can actually test in the lab by just kicking up the temperature on the incubator that we grow these cells in, instead of keeping them at a normal 37°C, which is body temperature. Maybe kick it up a bit to what it would be on a summer, hot summer day, and see if it causes the problem to be worse. So that could be why. That's a hypothesis, but I think that's been one of the reasons that's been thought.
Yeah. Okay. We've had a couple of questions more come in.
Sure.
Dupilumab. We know dupilumab has been tested for various EB types. Do you know if it's been tested on EB simplex patients?
So my
in the US?
understanding is, my understanding is there is a trial for EB. I can't remember the exact enrollment details, but I think Amy Pollar's group had put in a trial to do dupilumab, and I think EB simplex patients were in that trial, but I'd have to check that to be sure. The goal there is to say a lot of EB patients do suffer with itch, and dupilumab has really shown a remarkable effect on itch. So if you use it for atopic dermatitis, it makes a remarkable difference in children who have itch and can't sleep. It might be, I think, more treating the symptoms of the disease than the primary pathology of the disease. But if that makes a difference for the community, they should have access to that therapy. We just have to show that it actually works, which is why these great investigators are actually doing trials for it, which is fantastic.
Okay. Another question. Somebody noticed your differentiation drive. Sorry, your interesting remark on the differentiation drive in the models. What do you think about EGFR inhibitors as potential stimulus for differentiation and therefore therapeutic effects?
Yeah, so this is something we're very interested in, and there have been sort of attempts at using EGFR inhibitors to promote keratinocyte differentiation. So that's really where our logic comes from. We've just gone downstream a little bit, a couple steps down from EGFR, and are really looking at MEK inhibitors, which are a different way to target that same pathway. But I think the commonality is that they do drive keratinocytes to differentiate or to mature a bit more quickly, which may mean that they express keratin 1 and 10 more quickly, which we think might help with the pathology to make the cell stronger. But right on, that's exactly our logic, is that by targeting that pathway or downstream steps in that pathway, we might be pushing the cells to differentiate, which again is going to have a therapeutic window where you don't want to go too far. And there are some toxicities associated with these things. They're not perfect drugs. But I think for many patients with severe EB, they would be willing to endure some amount of side effects for the potential benefit that some of these therapies could have. But of course, there has to be safety studies done to make sure these things are safe in our patients too.
Yeah. Just a quick follow-up question for me here. So, for example, your MEK inhibitor, which is used in cancer, do cancer patients have stronger skin as one of the side effects of using it?
So what they tend to get is acne, which is interesting. And one of my theories, and again, this is just me guessing, right, is that, one of the ways that acne probably forms is that the keratinocyte differentiation may be happening in a dysregulated manner. The cells might actually stick together too strongly. And so then the normal sort of turnover of the skin might not happen in the way that it does. And that may be why their pores sort of get or into an acne, their glands, sebaceous glands get plugged up, and maybe that's why they get acne. That's me guessing. But maybe that's why. That would be one possible reason. But I don't think anybody has done any sort of like frictional test to make sure their skin is stronger. The other thing is I think there's a therapeutic window there if you have overactivation of a pathway. In the EB simplex, we may see overactivation. So if we can just get it back down to a normal level, we're not looking to shut it all the way off. That's probably not a good thing. The other thing is in cancer therapies, often we use a high dose because we don't want to give any chance that those cancer cells keep growing. So they often use a much higher dose than we might need to use in a disease like EB simplex, where we're just kind of trying to bring it down to the normal level of signaling through those pathways rather than shutting it off completely. So I think there's some room there to dose titrate down to reduce the side effects that might happen from those drugs. So that's a great point.
Perfect. I don't think there's any more questions coming through. And I think we're almost close to 9 o'clock. So I think
wonderful.
So on that note, I think we'll close the Q&A session there. And just on behalf of everybody, I just want to say really a huge thank you. It's been absolutely a riveting talk. I've learned so much. The questions coming through have been really stimulating, and the discussion's been really, really good. So I want to say a huge thank you.
Thank you.
Before you go, I'd like to remind everybody of, I wonder if you can see that. Sorry.
Yeah,
Or,
now I can see it.
oh, now you can see it. Let me close the question down. Sorry. So our next webinar is in December. And this time it's by Professor Bishop Omari, who's also talking about therapeutic approaches to treating EB simplex. So again, please do register. But before I close the webinar, again, I would like to thank you, Cory, for another amazing webinar. Thank you for all your help and all your work on helping the EB simplex community. So thank you for that.
Thank you very much.
Thank you.
Appreciate the chance to talk to your community today.